production technology and related fields. Currently, there are more than 60 participating institutions nationwide. The network organizes the annual meeting of the Thai Animal Science Network every year, and this marked the 15 years of the conference. The Department of Animal Science and Feed Technology from Faculty of Agriculture, Natural Resources and Environment, Naresuan University has the honor of being the main host for the conference. ภาคีเครือข่ายสัตวศาสตร์แห่งประเทศไทยนะคะนับได้ว่าเป็นความร่วมมือระหว่างสถาบันการศึกษาของประเทศไทยที่มีการเรียนการสอนทางด้านสัตวศาสตร์สัตวบาลเทคโนโลยีการผลิตสัตว์และสาขาที่เกี่ยวข้องซึ่งปัจจุบันค่ะมีมากกว่า60สถาบันทั่วประเทศและมีการจัดประชุมภาคีเครือข่ายสัตวศาสตร์แห่งประเทศไทยเป็นประจำทุกปีโดยปีนี้เป็นการประชุมครั้งที่15ซึ่งสาขาวิชาสัตวศาสตร์และเทคโนโลยีอาหารสัตว์คณะเกษตรศาสตร์ทรัพยากรธรรมชาติและสิ่งแวดล้อมมหาวิทยาลัยนเรศวรได้รับเกียรติเป็นเจ้าภาพหลักในการประชุมThe Animal Science Association of Thailand, under the loyal patronage of Her Royal Highness Princess Mahachakri Sirinthorn, was established in 1976. The objectives are to serve as a platform for disseminating information, exchanging knowledge, sharing academic experiences, fostering collaboration, providing assistance and support to its members, and also support the academic departments, educational sector related to animal production throughout the country. Currently, Mr. Suthep Wongruan holds the position of President of Association. สมาคมสัตวบาลแห่งประเทศไทยในพระราชูปถัมภ์สมเด็จพระเทพรัตนราชสุดาสยามบรมราชกุมารีได้ก่อตั้งขึ้นตั้งแต่ปีพุทธศักราช 2519 โดยมีวัตถุประสงค์เพื่อเป็นสื่อกลางในการแพร่ข้อมูลแลกเปลี่ยนความรู้ด้านวิชาการประสบการณ์รวมทั้งการสร้างความร่วมมือการให้ความสนับสนุนช่วยเหลือด้านต่างๆแก่สมาชิกชมรมสหกรณ์ผู้เลี้ยงสัตว์ทั่วประเทศและการสนับสนุนภาควิชาการภาคการศึกษาที่เกี่ยวข้องกับการผลิตสัตว์ทั่วประเทศค่ะโดยปัจจุบันมีคุณสุเทพวงศ์รื่นดำรงตำแหน่งนายกสมาคมThis year, the Department of Animal Science and Feed Technology, Faculty of Agriculture, Naresuan University, has the honor of being the main host of the event in collaboration with 4 lower northern regional institutions. These institutions include Rashmangala University of Technology, Lanna, Pisnulok, Ratchapat Pibun Songkhram University, Majo University, Prague Campus, and Phayao University. Additionally, educational institutions from across the country are also participating as co-hosts, providing support in terms of budget, personnel, and researchers to present their research work at the conference. More than 50 institutions and about 34 private sectors are supporting the conference financially. The event is expected to attract around 350 participants, including researchers, scientists, government and private sector, as well as the students and interested individuals. There will also be private sector booths to showcase the potential of Thailand's animal industry, as you can see. ปีนี้นะคะสาขาสัตวศาสตร์และเทคโนโลยีอาหารสัตว์คณะเกษตรศาสตร์มหาวิทยาลัยนเรศวรก็ได้รับเกียรติเป็นเจ้าภาพหลักในการดำเนินการจัดงานในปีนี้ค่ะแล้วก็ประกอบด้วยภาคีเครือข่ายนะคะหลายๆสถาบันโดยเฉพาะอย่างยิ่งภาคเหนือตอนล่าง4สถาบันนะคะที่ได้กล่าวไปแล้วค่ะ The objective of the event are to support and promote the exchange of knowledge and research presentation from faculty members and students in the field of animal science, animal husbandry, animal technology, and animal-related innovations. It aims to create a network of academic cooperation among educational institutions, the Thai Society of Animal Science, the Department of Livestock Development, and both public and private sector organizations. The focus is on the presenting of research in various fields. วัตถุประสงค์ของการจัดงานในวันนี้นะคะก็เพื่อการสนับสนุนแล้วก็ส่งเสริมการแลกเปลี่ยนเรียนรู้การนำเสนอผลงานวิจัยจากคณาจารย์แล้วก็นิสิตนะคะที่เกี่ยวข้องกับศาสตร์ทางด้านสัตวศาสตร์สัตวบาลเทคโนโลยีและนวัตกรรมทางสัตว์ค่ะหรือว่าสาขาที่เกี่ยวข้องเพื่อสร้างเครือข่ายความร่วมมือทางวิชาการแก่กันค่ะตอนนี้ก็คิดว่าน่าจะได้เวลาแล้วนะคะ Before we begin the morning sessions,It's an auspicious event that we are honored to have Professor Dr. Juan J. Loor as a keynote speaker. Allow me to introduce a little bit about him. He got his PhD in animal science from Virginia Tech, Blackbirds, and he's currently the professor of animal science and Division of Nutritional Science, Department of Animal Science at the University of Illinois, Urbana-Champaig. He has got a dozen of outstanding achievements with a number of publications, around 18,779 citations with 68 H index. Please welcome him on the stage. Now, I would like to invite Professor Dr. Juan J. Loor on the stage, please. Today, he will deliver a speech in the topic of nutrigenomics and climate change. What are the opportunities? Please give him a big round of applause. Thank you.
Well, thank you very much for the nice introduction. First, I'd like to thank the organizing committee for this great opportunity, a chance to see old friends and make new friends. It's been very nice. You really have a beautiful country, beautiful people, very friendly, and it's my pleasure to be here. So today I'm going to talk a little bit about, there's really three sections to this presentation. The first one has to do with just stepping back a little bit and thinking about why climate change is important, not only in industrialized countries like the USA where I work, but especially in developing countries like Thailand and other Southeast Asian countries. So I'll talk a little bit about that. I will focus primarily on dairy nutrition because that's my main area of expertise and really focus. And then we'll get into how some of the use of genomics tools are going to help us make progress, be more efficient in terms of managing livestock, particularly ruminant animals. And I'll show some examples of what's already been done. So I will end the presentation talking a little bit about where I think we should be going in the context of using nutrition and really understanding the molecular regulation in the context of heat stress and managing animals. Okay, so this review that I show here, it's actually very unique because it highlights some of the work that has been done in the context of understanding the effect of climate change. And again, climate change is not only heat stress, it's not only high humidity, drought. Climate change is really a combination of many different factors, but the interesting thing is that heat stress is really the one that has been studied the most, probably for good reason. And if you can see here, in the last 20 years, we've put a lot of emphasis in trying to understand the role of heat stress on livestock production. Not only ruminants, you can see here on the number of papers that were used for this review, most of it was with ruminants. Why? Because there are some inefficiencies in the rumen because of emission and production of some gases, methane, CO2, etc. But there are other livestock species that obviously also contribute to livestock production, pigs, poultry, etc. So there's been a lot of interest in the last 20 years. There's going to continue to be interest because the climate is changing. Things are getting a little bit more challenging. The other thing that I wanted to highlight before we get into more of the science that I want you to be aware of is the fact that we've known for many years what are the basic physiological impacts of something like heat stress or increases in thermal environmental temperature, okay? Decrease in feed intake, that's the classical response. Of course, the animal is trying to dissipate heat at whatever cost. But that triggers many different responses in the animal. We'll talk about a few of those that we can potentially change or affect through nutrigenomics. Okay, and of course, there's some other physiological events that happen in the animal due to this increase in temperature. That direct impact of heat stress on the animal has unintended consequences, which here are listed as indirect impact. Okay, and again, you can go through this list. So I'm not going to talk much about non-animal related aspects because it's not my area of expertise, but I do want you to be aware as animal scientists that we don't raise animals in isolation. We have to feed them. We have to make sure they have access to clean water. So everything it's related, and at some point I think we're going to have to talk a little bit more with our crop scientists, plant scientists to try to work together and mitigate some of these issues. Now let's talk a little bit about the physiology or the main aspects, physiological aspects that happen in the animal in the context of increasing temperatures. Now, I will like to highlight that depending on the species of animal that you're thinking about, some are less susceptible and some are more susceptible to slight changes in temperature. Pigs, for example, and poultry, they're very sensitive to small changes, smaller increases in ambient temperature. Ruminants can tolerate a little bit better, and of course, small ruminants like goats are really good at tolerating high temperatures. Some of that has to do with genetics. Okay, there's breeds of goats, for example, that can tolerate heat better than others. Also, cattle. I won't talk too much about the genetics of that, but I want to emphasize that a decrease in feed intake because of an increase in environmental temperature is going to trigger a cascade of events related to energy balance in the animal. Just to get rid of this excess heat, the animal is going to have to divert energy towards those processes. So maintenance energy use is going to increase. That essentially means that productive purposes are going to be affected negatively. There's going to be less nutrients going towards milk production, less nutrients going to growth. So this is very well known. Okay, these effects at the level of energy metabolism are relatively well known. In the last 15 years, roughly, at least in the dairy cow, which I have shown here, we've learned a little bit more about other important physiological events that also are negatively impacted in the context of heat stress. As you can see here, I'm just going to emphasize a couple of things. In the cow that is heat stress, of course, feed intake is going to decrease very quickly, but there are some other physiological events like activation of the immune response, activation of systems that need to restore homeostasis. For example, permeability of the GI tract is going to be affected. Those animals are going to be more susceptible to potentially toxins moving from the GI tract, and not only the rumen, but also the small intestine, into the circulation, okay? And of course, since the animal is eating less, nutrients are going to be moved away from production, and they would have to go to maintain the animal alive, okay, essentially. And the immune cells are a very important component of that. So again, summarizing some of the events that happen not only in ruminants but also other species, feed intake goes down, immune function is negatively impacted, the permeability of the gut is going to be affected. Okay, there's going to be more, potentially more molecules that can cause an immune response moving into the circulation. The concept of leaky gut. It's one that's been studied fairly closely, at least in ruminants and dairy in particular, in the last 5 to 10 years. Again, we've done quite a bit of work, and we continue to do work in trying to alleviate this issue of gut permeability, especially from the immune standpoint, because, again, if the gut becomes more permeable, the animal is going to be faced with a number of molecules, some of which are going to trigger an immune response. That's why it's very common when you're doing these experiments or when you're going out to farms and potentially take a blood sample to see what's the physiological response of the animal during heat stress, you can measure more inflammatory markers in the circulation. You can measure more oxidative stress markers. And these two events, inflammation and oxidative stress, they're really linked together. Okay, so there's a vicious cycle between both of those. And as nutritionists, we're trying to understand how we can manage the animal during heat stress, for example, in the summer months, in the context of potentially some bioactive compounds, potentially some functional nutrients, which I'm going to talk about in a little bit. So, again, all these processes that happen in the animal from the time that the intake of feed is reduced are going to impact the use of nutrients for productive purposes. This is just the reality, not only in dairy cows, but also across different species. Okay? In the long term, of course, if the animal is in negative energy balance, if the animal cannot consume enough nutrients, not only production is going to be affected, but potentially the ability of the animal to become pregnant again. Okay? So there's an important cascade of events that we need to keep in mind when it comes to understanding the effect of climate change and heat stress, in particular in the animal. Okay, so now with that in mind, you might be thinking, okay, we know that the temperature is increasing gradually, it's going to continue to increase. Is livestock production going to be affected negatively? In short, yes. And some of that not only has to do with trying to control the animal in some way so that the animal is able to produce optimally during these changes in climate, but also in terms of the public perception. This slide that you see here was taken just last week from a news magazine, CNN. Some of you might be familiar with it, and you can see here that there is there's an important number of the public in the United States in particular. There's some of this movement also in Europe that are trying to figure out whether we can generate meat in this particular example without actually raising livestock. There is a sector of the population that believes in this type of approach. Of course, as livestock scientists, we understand that raising animals for food, it's not only about providing nutrition to humans, but it's also about livelihoods, especially in rural communities. So what I'm showing you here should make us aware that we have to really think carefully about how we're going to continue managing animals, especially in non-industrialized countries, in developing countries. In the U.S. and some parts of Europe, we're going to continue to feed high-quality forage because we have it. Okay, we have high-quality hay, for example, in the U.S. We have plenty of corn silage in the Midwest. We have plenty of soybean meal. So for us in the U.S., we're going to continue doing that. But you're also going to see the industry trying to position themselves to try to, again, provide not only specific nutrients, potentially some feed additives, to try to help optimize the response in the animal, to try to also help mitigate some of the issues related to climate change. And I'm showing three important aspects of managing dairy cows: fatty acid nutrition, amino acid nutrition, and ruminal digestion. Okay, so again, these are things that we can do in the U.S. and some European countries. We're also starting to look more closely at some of these nutrients in the context of functionality, meaning are some of these nutrients going beyond just serving as building blocks for lipid, building blocks for protein? And I'll talk a little bit more about that. In fact, in fact, I think there is a great opportunity, especially in emerging economies like Thailand and other Southeast Asian countries. I got these numbers from a website just to try to illustrate in the context of dairy production. China has been growing immensely in the last 20 years. They made a conscious decision that they needed to provide the growing population a nutritious food, and milk obviously was an important aspect in that context. And the number of dairy cattle has been growing exponentially in China over the last 20 years. Of course, China has other issues. They don't have good quality forage. They have to get it from the U.S. primarily, and they have some issues with environmental pollution as well. But again, this is the reality that they have in China. One of the things that the Chinese, I believe, have been very keen on is in promoting scientific research in livestock production, and particularly dairy production. They've invested a lot of funding, and I know that because I've been collaborating with a lot of Chinese colleagues on the northeast of China for about 15 years. And again, that's another aspect that comes into play when we're thinking about how, for example, dairy production is going to keep moving forward. But in the context of livestock production, we also need to think about other important species. So the growing population is going to have an impact on that. There's going to be more need to potentially feed higher quality feeds to animals. Is there a role, for example, for some agro-industrial byproducts? And I know in Thailand there's been quite a bit of research, so I did a little bit of searching, and I'm showing just a few examples of the work that has been done here in Thailand to try to address primarily issues related with ruminal digestion. Okay, and I list some of the main results that have been obtained. Some of these byproducts are obviously going to impact what happens in the rumen, not only the bacteria, and they potentially do that through the actual bioactive molecules, okay? So I think there's a lot of room to keep understanding what are the bioactive molecules in these different products that you generate here in Thailand in the context of a physiological response. And I have a list of things, okay, physiological outcomes that have been generated, okay, by feeding or supplementing a number of these different byproducts. So I think there's a lot of opportunities. There's been some research also in non-ruminant species, and I wanted to highlight this work with poultry because it also went a little bit further in the context of understanding potential molecular regulation. What I think it's still missing a little bit is understanding the mechanisms, okay? And in order to do that, we need to look at the rumen itself, for example. We need to be able to harvest the papillae. We need to be able to look at metabolites being produced, and we need to be able to connect those two, okay? And I'll talk a little bit more about how we've tried to achieve some of those processes. And when you think about the regulation of biology, of course, it happens at different levels. I showed you a few things. This field of nutrigenomics or nutritional genomics, however you want to call it, it's not new. Okay, the first paper that kind of summarized the concept was published in 2003, so about 20 years ago. Okay, and it's a very simple definition or concept. Some nutrients can actually trigger a functional change in an organ through its interaction with some molecule, okay, a protein, for example. And again, there's a number of nutrients that could potentially do that. I will show you some examples. But in the context of the scientific framework, I want to show this slide because it really highlights the role that we as scientists in the animal production space can have. When we started doing a lot of this genomics work, we were working just looking at gene expression in the context of some nutritional management. And you see this at the bottom of this, on the right of your slide. Again, that's one of the first things that you can do is just profile your tissue of interest in the context of a particular nutritional treatment. You generate data, okay? Then you need to analyze those data. You need to find the proper approach. You're going to hear the term bioinformatics analysis, and that's essentially what that field is supposed to be. We've done quite a bit of feeding studies. I've done a little bit of work with pigs as well, mostly cattle, beef, and dairy. And what we're able to do is not only look at the phenotypic outcomes of these nutritional management approaches that we use, but also look at the cellular responses. And that has allowed us to, again, generate more hypotheses that we can then test. And I'm going to talk a little bit about a specific example that we have used. In the context of nutrition and changes in gene expression, we need to remember that there are specific proteins referred to as transcription regulators that will be important in the context of something like an amino acid, something like a fatty acid, changing the expression of certain genes. And the bovine genome has quite a few of these so-called transcription regulators. This is from a paper that did a pretty comprehensive analysis. You can see here there were over 700 genes that potentially encode a transcription regulator. Not all of those are susceptible to a nutrient, but many are. The other thing to keep in mind is that for the bovine, we don't know what most of those do. Functionally, we do not know what they do. So again, there is opportunity to try to understand that a little bit better. And there's also opportunities to try to understand in which tissues and for what physiological function these transcription regulators are very important. Now, in our lab, we've worked quite a bit with a category of transcription regulators that is referred to as nuclear receptors. As the name implies, these proteins are bound to a DNA sequence in the nucleus, meaning that changes in nutrients, and again, it could be fatty acids, it could be amino acids, it could be trace minerals, that's what's going to trigger activation of the response. In this example that I have here, I'm showing the classical PPAR, RXR protein complex. PPARs respond to fatty acids in particular, so when fatty acids, certain fatty acids increase, that's going to create or cause an activation, which you have at the bottom here, and that will trigger changes in gene expression, some of which are also going to translate into more protein in the cell. And again, a lot of these proteins are enzymes, so capacity of a particular biochemical pathway could potentially increase. Gluconeogenesis, for example, adipogenesis, okay? So there's a lot of opportunity to try to understand what these different transcription regulators do. I'm showing two examples of important transcriptional and translational regulators, because even though we talk about nutrigenomics, we have to keep in mind that regulation, at least of metabolism, not only occurs at the transcriptional level, but we now know that there are proteins and signaling pathways that also respond to nutrients, but changes occur at the post-transcriptional level, okay? And that's the case for mTOR, okay? And there's been quite a bit of work done with livestock species, particularly ruminants, in the context of understanding how these different molecules or signaling pathways work. One of the first things that we had to do before we got to this point was to try to understand where these different networks are located in terms of abundance. Is the liver an organ that can potentially be regulated by some of these PPARs or mTOR? Is the mammary gland? Is the adipose tissue? Okay, we've done quite a bit of work with that. And again, I indicated before, in order to make sense of a lot of these data that you generate, you have to use bioinformatics, and this is the approach that we typically use. We look quite a bit about, when it comes to this analysis, to pathways. Again, all biological pathways that you can think of. There are some databases that are very comprehensive. We also do quite a bit of gene network analysis. And again, to do gene network analysis, there's some commercial software that you can use, but there's also some freeware that you can use to try to do a little bit of this analysis. I think moving forward in agriculture, we're going to see more development of tools that are freely available. And those are the ones that I show at the bottom. Freely available in the context of you being able to upload your data and generate some meaningful biological output. So that's an important area of research. Now let's talk a little bit about how we in our lab have used these approaches to try to understand what happens in some key tissues of the heat-stressed dairy cow. We do a lot of bioinformatics analysis. This is just to highlight how you can take publicly available data. Oftentimes when people need to publish papers dealing with sequencing data, they're required to deposit the actual data in a public repository so that anybody that wants to use it can do that. And what we've done is taken some of this data that's freely available and used bioinformatics to try to understand what happens in the heat-stressed tissue, particularly the small intestine and the rumen. We're very interested in understanding what's going on there, but we've also done it with the mammary gland and with the liver, which are obviously important metabolic tissues. Now, why are we interested in the gut in the context of heat stress? As I indicated before, there is a lot of evidence indicating that heat stress is going to increase permeability of the GI tract. Historically, most of that work was with the intestine, but in the last 3 years, there's been more evidence that the rumen is also going through some of these issues. For example, heat-stressed animals are going to have a lower density of villi, which obviously are very important for absorption. And in fact, what I want to highlight is that not only heat stress causes an important effect at the level of the gut, but also, for example, limited access to feed, okay? Maybe we forgot to give enough feed to all the animals that we have. You have some behavioral aspects. Some of the older cows might, be able to eat what they need, but maybe the younger heifers are not going to be able to do that. So the gut, it's very important in the context of stressors, and that's why we're very interested in trying to understand how those tissues respond. This is just a pipeline that we use when we're doing this type of analysis. Again, taking data that's publicly available. We do statistical analysis, so we essentially have 3 different steps before we can actually use a software that was developed by one of my students to try to look at different gene networks, okay? So the beauty of this type of analysis is that we don't have to go and run an experiment that is going to take 1.5 years. Okay, as long as we have access to data, we can start doing data mining. We can start looking at affected pathways. This is an example of an important pathway that was affected in the liver of cows that were calving in the summer. So we're comparing the liver of the cow calving in the summer relative to the spring. The one-carbon metabolism pathway actually has become very important in the context of the focus in our lab, simply because it seems to be very important under many different conditions, and specifically in the context of heat stress. So all the arrows that you see here indicate that there were changes in expression of all these genes that we're listing here in this box, okay? For those of you who are unfamiliar with the one-carbon metabolism pathway, this is an abbreviated form of the entire pathway. I want to highlight a couple of things. One, that the one-carbon metabolism pathway is susceptible to nutrient supply. Methionine, choline, betaine, for example, and a number of non-essential amino acids, because they complete a lot of these different pathways. The other important aspect of this one-carbon metabolism is that water-soluble vitamins are also an important component in the context of being cofactors for some key enzymes. So we worked on this area of research for probably more than 10 years, trying to understand primarily the role of methionine. Again, methionine is an essential amino acid, as we know, but we recognize that methionine, because of its impact on this one-carbon metabolism pathway, it could potentially also serve as a functional nutrient, meaning, for example, that methionine flux through the methionine cycle could help generate glutathione and taurine, which are important cellular antioxidants. And in fact, that's how we started working in this area about 15 years ago. Now, one-carbon metabolism is not only important in the context of antioxidants. Again, if you think about the most basic biology, DNA replication, okay, is a very fundamental process. For example, here in ruminal papillae of heat-stressed cows, a lot of these components of the DNA replication pathway were upregulated, and that's what the red color means. You also see a lot of green, meaning that a lot of other components associated with this overall process were actually downregulated. Okay, so this is how we can use this type of analysis to try to understand what's going on. So the rumen is actually going through a process in which potentially heat stress is causing a situation in which cells need to be replenished, meaning that they're going to need more nutrients. They're going to need to potentially regulate blood flow to the gut, which could potentially be a challenge, especially when the animal is trying to dissipate heat. In the context of metabolism, I want to highlight the impact of heat stress at the ruminal epithelium in the context of amino acids, and the fact that a lot of these pathways that you see here are connected in some fashion with one-carbon metabolism. Remember, one-carbon metabolism is not only about methionine, choline, betaine. There are a number of non-essential amino acids that complete these three different branches of the pathway. And one of them, for example, the one related to glutathione synthesis, it's also very important. Now, glutathione synthesis, it's important for reduction of peroxide radicals, like hydrogen peroxide. And again, an enzyme like glutathione peroxidase not only requires glutathione, but also requires selenium. So again, another example of how this pathway really it's one that requires a lot of coordination among many different nutrients. Now, I do want to point out that some of this work that has been done in the context of one-carbon metabolism in ruminants, it's also pertinent to other species. In this case, for example, there's been quite a bit of interest on using betaine. Again, betaine is the oxidation product of choline. Okay, so any tissue in birds, for example, that has one-carbon metabolism activity could potentially use choline to generate betaine. And betaine can then go through these pathways that I showed you before and potentially help alleviate a lot of these different functions that we know happen in the heat-stressed animal. Now, again, our focus has been in the lab on methionine. We're starting to work a little bit outside methionine and looking at other potential important nutrients within this one-carbon metabolism. We've been working since about 2008 on this topic. We published quite a bit in this area of methionine and one-carbon metabolism in the context of production in transition cows. Again, the transition period being very challenging metabolically and physiologically. And we've been able to this is going the opposite way. It's going the opposite way. I don't. Oh, wow. Okay. Yeah, a few slides. Oh, that way, okay. Yeah, this remote is not, I'm not used to this type of remote, so I will. Yeah, we're getting close. Okay, let me see if. Okay, so there's a slide that is missing, but what I wanted to highlight was that using the type of bioinformatics approach that I showed before, we've done quite a, we tried to identify networks, as you can see here. The one on the left of your screen is a liver network that we built. This paper was published back in 2014, and what we were trying to do was to use bioinformatics and network analysis to understand linkages between some of these important transcription regulators and a number of genes that were affected when we were overfeeding energy to cows in the dry period. So that was our nutritional approach. I wanted to highlight this transcription regulator because this is very important in the context of the antioxidant response. So the abundance or the activation of this transcription regulator, which happens primarily at the post-transcriptional level through phosphorylation and dephosphorylation, it's important because there are a number of nutrients that we now know are potentially triggering a response in the animal. Okay, I'll talk a little bit more about that. This other PPAR is also important, primarily at the level of the adipose tissue, and in some immune cells because it's going to control nutrient use. In the case of the adipose glucose, for example, it can also have an anti-inflammatory effect. Okay, so again, we've used, you can, the beauty of using bioinformatics approaches is that you can do all of this data mining to try to understand what's in these tissues that you didn't expect to see potentially or that you didn't know existed. And going to this, specifically about this antioxidant response and this PPAR response, I wanted to highlight the potential role of some of these bioactive compounds that fall into the category of polyphenols. And you can see quite a few different molecules. I know there's been quite a bit of work here trying to identify some of these bioactives. I think there's a lot of potential to continue to fine-tune which byproducts potentially provide more of these to the animal. What I think needs to be done as a next step is to try to understand at the molecular level what some of these byproducts potentially are doing in the tissue. I think that's something that potentially can be done in the future. This question about the use of byproducts is not only relevant to Thailand, it's not only relevant to some countries in Southeast Asia, but countries in other parts of the world. Brazil, Spain, Australia. They generate a lot of agricultural byproducts, and people are starting to try to figure out how they can be used to feed livestock. Okay, so these are important questions throughout the world. Okay, so that's an important aspect to keep in mind. Coming to the end of my presentation, I want to close emphasizing that there's a lot of work in the U.S. in particular trying to manage heat stress in ruminants, beef, and dairy. We don't rely a lot on agricultural byproducts. Again, simply because we are fortunate to have good high-quality forage, plenty of corn, plenty of soybeans, which again provide the main nutrients, the macronutrients. That doesn't mean that we couldn't potentially supplement some unique byproducts to try to enhance the availability of some of these key bioactives. I go back to betaine, for example. And there's some work indicating that potentially certain byproducts with high betaine content can have a positive impact in the cow during heat stress. The last thing I want to say about the so-called nutrigenomics effect is that we need to think about what's happening in the GI tract, what's happening to those microorganisms, especially in the ruminant animal, what are the metabolites produced and how they can impact not only the ruminal epithelium, but potentially signals that go in, to the animal and potentially regulate feed intake. That's an important aspect of what I think we need to do. Now, moving forward, there's going to be a balance, okay, between climate change and livestock production. That I think is evident. It has been obvious for many years, and that's the key. How do you maintain that balance? Okay, we're going to have to continue trying to understand how nutritional management helps in terms of getting these animals to adapt in an optimal fashion to changes in water availability, increases in temperature. I think that's where we animal scientists are going to be very important. But again, there has to be some sort of communication with other scientists in the agricultural fields, crop sciences. So it's going to be a challenge, but I think we can certainly make strides if we really apply ourselves into studying more those topics. Of course, I'm going to end emphasizing that international collaborations are very important. I've been fortunate to collaborate with some of you in this room, and I want to say that it's been a great experience for me working with some students. I've seen some of you here. What is Krong? That's Tony down there. It's been a great experience for me. Very talented students, hardworking students, and as you can see, they've been able to generate important data that has helped move our program forward. Okay, and I'm hoping that we can continue these types of collaborations in the future. I want to highlight my other Southeast Asian colleague from Canto University in Vietnam. He was a Fulbright scholar in my lab, and that's another type of opportunity that I think could be used by some of you in this audience. And with that, I appreciate your help and apologize for the technical issues.
Thank you so much. That's such a wonderful and interesting speech. May I take this opportunity to invite Assistant Professor Dr. Warasit Thojampa, the Dean of Faculty of Agriculture, Natural Resources, and Environment on the stage to give a token of appreciation to our keynote speaker. All right. Some question from the stage, from the floor. Sorry, there's one question from the floor.
Thank you very much, Professor Loor, for such a very wonderful and comprehensive lecture this morning. I think all of us have been enjoying a lot. And you have given us such a wealth of knowledge on the very hot issue that everyone has been interested in, carrying out the research. And I also wish to thank you for coming here. You've been helping a lot of Thai scientists, not only Thai scientists, but the global scientists as well. Among many interesting issues that you have addressed this morning was the issue of nutrigenomics and the heat stress, right? But when it comes to the applied aspect of manipulating the dietary regimes, would you care to elaborate a little more on the practical point of view on this aspect, especially the use of polyphenols that we have abundantly in Thailand, and we can go forward, and also the importance of the nutrigenomics on the role of the peroxisome proliferated activator receptors on that into the function of food. That's what I'm trying to get to. in the milk. That would be very useful. Thanks a lot.
Great. Thank you, Professor Mehta. Great question. So 2 questions. The first question, how do we apply this in the field? I will tell you our experience with methionine. Okay, I'll start there, and then I'll say a few things about how maybe some of these byproducts can be used here. But when we started working with methionine, again, with this idea that we could enhance antioxidant capacity, we really didn't know, how much more methionine these animals required. We knew that lysine and methionine are the top 2 essential amino acids for dairy cows in established lactation. So when we started thinking about, okay, shall we double the supply of methionine during the dry period and then the transition? Is that going to give us what we want? So we had to go into that project, trying to use informed guesses. Okay, we were successful in the context of increasing methionine supply. Probably 30% relative to what cows were typically fed at that time. That's 15 years ago, okay? So by doing that, we were able to show that more of these essential amino acid was able to have a positive impact on the dairy cow. Cows ate better. They adjusted to lactation faster. They produced more milk, so efficiency went up. In addition to that, the inflammatory state was reduced. Okay, and we've done a lot of work showing how the increase in methionine supply also led to an increase in glutathione. We've measured some enzyme activities in that pathway. So farmers now in the U.S., my colleagues in the industry tell me that they have implemented these suggestions. So now they're increasing the supply of methionine during the dry period and during transition. So that's essentially they're feeding more, okay? But they have some guidelines. Now, when it comes to some of the byproducts, that's a little more challenging, at least for me, because I don't work in that area. I did read some of the papers that I cited, and it wasn't clear to me how these byproducts are being added in the diet, in the context of, okay, should we feed 1 kilogram or half a kilogram? That wasn't clear to me, but I think one of the things that probably needs to be done, in case that it hasn't been done, is look at a titration potentially of some of these feedstuffs. Understanding what's in some of these feedstuffs from the bioactive standpoint, polyphenols, if this is what you're interested in, understanding that profile, doing these titrations, and having a clear physiological outcome, antioxidant capacity, inflammatory markers, okay? I think that can be done, but before you get to that, unless you do that, I'm not sure that you can really go and tell the farmer, okay, you need to feed 1 kilogram. Maybe it worked in one experiment, but maybe she can use 2 kilograms of this product.
Yes, let me just share a little bit here. We have been working on using these byproducts from fruit, which contains very high polyphenols compounds. Thereby pelleting, this product into different ratios and supplemented about 1 to 2% of dry matter intake per day. That has resulted significantly, favorable in regulating or manipulating the rumen ecology, especially on the microbes there. So, this area will receive more. I believe that will be receiving more interest and into the application of the information and the knowledge. So I think that we will emphasize more, and of course we need your support. It has been great, and we will count on you, because there are many young scientists here have been interested in this area, especially in Nutrigenomics. So please help us moving on into the near future. Thanks a lot again.
It'll be my pleasure, as always. It's been great. Thank you. And I appreciate everybody here, very friendly, and you have a beautiful country, beautiful people. Thank you very much.
Thank you so much. So may I proceed to the presentation of the token of appreciation by Assistant Professor Dr. Warasit Thojampa, the Dean of Faculty of Agriculture, Natural Resources and Environment และขอเรียนเชิญแขกท่านผู้มีเกียรติบริเวณด้านหน้านะคะได้ขึ้นมาบนเวทีเพื่อร่วมถ่ายรูปร่วมกันค่ะขอเรียนเชิญค่ะขอเชิญท่านผู้บริหารนะคะได้ขึ้นมาบนเวทีแล้วก็ถ่ายรูปค่ะขอเชิญท่านรองศาสตราจารย์ดร.ทศพร อินเจริญนะคะรองศาสตราจารย์ดร.วันดี ทาตระกูลร่วมถ่ายรูปด้วยค่ะ Then we move to the group photo.ขอเรียนเชิญค่ะ After this session, there will be a research presentation on the ground floor and including this room. ต่อไปจะเป็นการนำเสนอผลงานวิชาการภาคบรรยายนะคะมีทั้งที่ห้องนี้ห้องมหาราช1นะคะสาขาโภชนาศาสตร์สัตว์เขียวเอื้องค่ะห้องที่2ห้องประชุมมหามนตรีสาขาพันธุศาสตร์และการปรับปรุงพันธุ์สัตว์ผลิตสัตว์เศรษฐกิจและสัตว์เลี้ยงค่ะห้องนำเสนอที่3นะคะห้องประชุมเทพนรงค์ลือชัยสาขาโภชนาศาสตร์สัตว์กระเพาะเดี่ยวและห้องนำเสนอที่4ห้องประชุมศรีหราชเดโชชัยงานวิจัยระดับปริญญาตรีค่ะเป็นการเก็บภาพความประทับใจนะคะค่ะขอมินิฮาร์ทหน่อยนะคะมินิฮาร์ท please โอเคค่ะ thank you so much ขอขอบพระคุณทุกท่านมากค่ะ thank you so much for the wonderful speech